Hello, it is still Sean Calingayan, your host for this two-part lecture series on forensic proteomics. You are now watching part two of the series, so if you haven't already, please go watch the first part as it sets a foundation for the content introduced in this lecture. Please note that this lecture series serves as an introduction to the world of proteomics and may not cover in-depth details regarding this discipline. Thank you for watching! In the previous lecture, we covered what proteomics is and how to conduct proteomic analysis. This half of the lecture will cover three applications of forensic science, some logistics, and finally a case study. Let's start off with the forensic applications. So proteomics is so diverse. Remember that was one of the advantages that we got for proteomics, that it was so diverse and that we could use it for anything, essentially anything with a protein, right? So let's think about what applications of forensics can proteomics be applied to. So I have two categories here. One is traditional forensics, traditional in quotations. So I refer to that as, I think of traditional forensics as crime scene investigations, like criminal investigations, things that are essentially not stereotypically forensics, but, things that are more commonly seen in like forensic context. So again, human investigations, death investigations, maybe environmental stuff, right? And stuff that is forensic related. So not really criminal, but still very important, like civilly. Is that word? Like so civil law and stuff surrounding civil law versus criminal law, and maybe in forensic related that can be also health and safety, so that's still forensic stuff, right? So take a minute, pause. I'm going to go over 6 examples, 3 in each side, and so just take a minute, pause the video, and think about it. You guys good? Okay. So I'm gonna go through them now. Some examples that I have is microbial forensics. Recall that proteomics was originally used to characterize microbial communities, so why not be able to apply that to microbial forensics, right? Wildlife identification, human identification, biofluid identification. So these are all your criminal investigations. So your crime scene, is this illegal poaching? What kind of animal is this from, right? And that is mostly for traditional forensics. Now moving on to forensics related. Sports doping. So we have sometimes athletes will dope themselves with testosterone or other hormones that are, protein. So protein hormones or protein steroids, stuff of that sort can be detected using proteomics. So this is where we would employ sports doping, forensic proteomics, excuse me. And then food investigation, so health and safety, if there's a protein toxin, let's say anthrax or ricin within a food supply, stuff like that. And finally, entomotoxicology and physical conditions. This is kind of both in forensic related and traditional forensics. This last point, entomotoxicology and physiological conditions, this can inform the pathologist or a pathologist if a death was natural or not. So think about SIDS. Recall SIDS. I'll talk about it again later, but recall that SIDS example. Did this infant die from negligence or was it SIDS? Think about it. So as many applications to forensics that proteomics has, I do want to focus on 3 specific ones for this lecture, mainly the ones that I find the most interesting. Number 1, human identification. So we're determining a person's DNA sequence from expressed proteins. Recall, this is why I kind of brought up the central dogma in the first part. We have proteins, we're going to reverse engineer it to determine a DNA sequence. Kind of cool. Number 2 is forensic investigations. There's, I think, 3 or 4 subsections under this one, which are fingerprints, cause of death, and postmortem interval. And finally, protein toxins, because who doesn't love a little protein toxin action, right? So the first topic I want to cover under application is human identification. So this essentially tells us all we need to know. There's a lot of words here, and I'm going to point them out to you. We have genetically variant peptides. So the GVPs, this essentially means that these peptides have a single amino acid polymorphism. So there's a single change in amino acids that is caused from a change in the DNA sequence. This change in the DNA sequence is called a non-synonymous single nucleotide polymorphism, or a SNP. And this image at the very bottom explains it beautifully. So on the left, we have this AGA in the DNA sequence, which causes an arginine, which codes for an arginine residue. In a mutated or a non-synonymous single polymorphism, or a SNP, we have an AGT now instead of an AGA. So that last A has switched to a T. This AGT then codes for a serine, and this causes a genetically variant peptide. Now that we've covered the basis on why proteins can be used for identifying humans, we can now look at how we do that with proteomics. So we've covered the workflow of proteomics already, and it's not too much different. The only thing to note here is that it's a targeted analysis search. So we recall that just means we set everything, all our instrumental conditions to get the mass spectrum of specific proteins that we know are in the sample. And that's what that green box at the top is. Those are just our targets that we that we look for within the protein sample. Now, the line at the bottom is very important because this bottom line says essentially, from the DNA, we're going to do our analysis of the DNA, and then we're going to translate that into protein, and then we're going to see if that matches up with the proteins present in the sample. That's what that means. So again, the proteomic aspect of it is not too different, or it's not that complicated. It is just a targeted proteomic search with the GVP targets. Yeah, with the GVP targets based on the suspect's DNA. Now, consider what challenges the analysts might have, right? So, number one, they need a suspect genome for reference/validation. How do we know that this protein that is expressed protein has expressed is not mutated? How do we know that's not the normal expression of this protein? Number two, same thing again, is this protein off or not? So we need to consider that sometimes different proteins from different tissues are expressed differently and may have modifications associated with them. And number three, statistical independence of underlying genes. This is more of a bioinformatics challenge, but it essentially just states that sometimes the proteins that are expressed may have been coded from genes that are not independent, which makes, the statistics and the calculation of error rates a little more complicated. Now, fingerprint analysis is the first part of the investigations topic. So fingerprint analysis, you are detecting proteins within a deposited fingerprint to get the spatial yield of the proteins. And you are typically using MALDI-MS for this. So recall MALDI, so matrix-assisted laser desorption ionization, is an ambient or independent technique, so making it more accessible for in situ proteomics. You are looking at the spatial yield of proteins within the fingerprint, and if the proteins and the peptides are different than expected or of different origin, that can implore you or indicate that you might want to develop the fingerprints and then infer the suspect's activity. So essentially with the fingerprint analysis, you're looking at proteins and trying to identify an origin or like a rough estimate of the origin. And if the origin and the fingerprint do not match, then that, indicates you should probably develop the fingerprint to see what's really happening, what's going on. There's a special case for certain fingerprints. Those fingerprints that are deposited in blood. You're detecting heme and hemoglobin subunits, which can differentiate blood from different sources, so non-human versus human sources of blood. And on the page at the very bottom, we also have to consider, there's something to consider for proteomics of fingerprints, is that most of the cells have cytokeratin. So 35% of the proteome, so one single cell, of all the proteins, they are cytokeratins. And these keratins inhibit the detection of forensically relevant proteins. And if this does happen, that just means you have to do extensive sample preparation to remove the keratins, and you just have to send it back to the lab. So that's what that non-situ methods are employed means. You just have to send them to the lab to do more sample prep. The next topic under investigations is the postmortem interval. We're using proteins such as alpha enolase, malate dehydrogenase, peroxyredoxin 2, and others such as GAPDH and EFF1A2 in skeletal tissue to predict how long ago a person died. That's essentially what it is. All these proteins that I listed, they do have a predictable half-life. So this is what predictable proteolytic patterns means. You could essentially predict how much died based on the concentration. And this is actually really highly effective in the early to mid postmortem phases. Important to note that the concentration and any valid end studies for postmortem interval have been validated via SDS-Page and Western blot experiments. If you're unfamiliar, SDS-Page and Western blot are just other ways to determine concentration of protein. The third application of proteomics in forensic investigations is the cause of death, which is only specific to drowning victims. So how you would do this is the cause of death is, measuring the protein levels from a sample collected from a deceased individual. And right now, the only research that has been proven to be effective, or proteomics to be effective for cause of death determination, is drowning. Because drowning victims show decreased alpha-1 antitrypsin and increased apolipoprotein in their system, or in their tissues when they drowned and died, unfortunately. And finally, the final application again is physiological conditions. Recall SIDS, as I mentioned at the beginning and I mentioned at the start of this this episode. Proteomics can inform the pathologist of physiological conditions determining nature or manner of death, right? So SIDS, decreased levels of 14-3-3 protein and tryptophan hydroxylase in infants indicates SIDS and not neglect. Various stuff like that. Final topic, the final application I'd like to cover is protein toxins. What you're doing with proteomics is you're characterizing these toxins within complex matrices using targeted ESI or multi-MS. Keep in mind, this is targeted because you know what toxin you're working with. You know what protein, you know the structure, hopefully, and you're just trying to isolate that and do your analysis and how much of it is in there, assuming that's the goal of the study. But first, you need to purify it, and this requires a specific type of purification. So this is immunoaffinity purification, where you're just using antibodies to extract the sample from the complex matrix, or from the protein from the matrix. Another thing you can do with proteomics and toxins is measure the level of activity. So one thing, this level of activity is specific to ricin. So this is the mechanism of how ricin works. It depurinates your DNA, which forms a free adenine, and therefore you can determine the change in adenine concentration using the proteomic workflow. But one thing with the measure of activity to consider is that ricin is not the only protein toxin that uses this mechanism. This mechanism belongs to the family of type 2 ribosome inactivating proteins, and therefore it lacks biological sensitivity. So, yes, you can measure the formation of free adenines, but that doesn't necessarily mean it's ricin specifically. It could be a different type 2 ribosome inactivating protein. That's just something to consider when measuring protein toxins and activity. So while introducing proteomics and discussing applications and of the such, I've mentioned here and there specific challenges that come up with proteomics specific to that work step. So recall the toxins where lacking biological sensitivity, the cause of death is only specific to drowning, and stuff like that. But there's still so much as like a general under the general umbrella of proteomics and forensic proteomics that needs to be discussed. The first thing is legal admissibility. I'm gonna pause because this is the Dober standard, and if you're watching this at an advanced level, I hope you know what the Dober standard is. So take a minute to remind yourself. If not, if this is your first introduction to the Dober standard, it goes something like this. It has a 5-point, essentially the Dober standard is a 5-point checklist, quote unquote. It's more so like a guide, or it's legally binding. So it is a checklist on whether scientific evidence is admissible or not in a court of law. And these 5 points in the checklist are general acceptance from the scientific community, peer review and publication of the method, rigorous validation and testing has been done on the method has standards and controls, and finally, error rates have been determined or can be estimated. For forensic proteomics, some of these are A-OK and some of them are more iffy, more ambiguous, right? Let's talk about it. With respect to forensic proteomics, general acceptance from the scientific community. Recall that it's been used for biological and biomedical sciences, so yes, it is generally accepted within the scientific community. Peer review and publication, not specific for for forensic proteomics, but again, proteomics has been published before and it has been peer reviewed before. Rigorous validation and testing has been done. Yes, but not really. This is why it's red. Red is no, because for forensic proteomics, it has not been validated, it has not been tested. So there's this issue with should we allow it because it has been tested and validated within the biomedical community and biological science community, but it hasn't been tested for forensic applications. So right now it's red, so it's not possible. Standards and controls, this is yellow because, well, yes, but also, well, no. Recall the heavy isotope method for determining absolute concentration where you have the internal standard. How often is that going to work? That hasn't been rigorously tested and validated. So that goes back to the point above it where we have standards and controls, but they haven't really been tested and validated, right? And finally, error rates can be determined or can be estimated. This actually, it has passed. There is something called the false detection rate, which is used to estimate and determine error rates. So let's move on to the statistics of it all. The error rate, which is associated with the false detection rate. This is a method for determining the score threshold. So recall the protein sequence match from the database search. You would have to score the compared databases and compared spectra, and then set a threshold for which is considered a true positive. And this False detection rate is essentially how you set the threshold. This is an expression of the proportion of false positives in the whole set, of tests, and not individual sets. This basically just says the false detection rate is basically saying from this set of positives, what is the proportion that are likely to be false positives? That's the whole point of the false detection rate. And this is why we consider this an error rate, because you're essentially accounting for things that might not be true positives. Important to note that the false detection rate is dependent on data set and the usage of the identification. The actual value of the false detection rate is not critical in forensics. Obviously, we want it to be as low as possible, but if it is high, it still fulfills the Dober standard criteria. Another thing important to note, I know I keep saying that things are important to note, but 1% is conventional for an FDR. So you want an FDR of about 1%, but there's obviously still research going on to improve the estimation tactics and methods of estimation for the false detection rate. Do we determine the false detection rate? Well, there's one method called the target decoy approach, where you're essentially having decoys, which are known false positives. So these are sequences that are not in the original sample. And then targets, which are the things that are the spectra that have been found, or the sequences found within the sample. And you're just going to take the ratio of false to over true and take that as your false detection rate. That's a pretty simple calculation. Remember, known false positives over true positives, essentially. Another challenge regarding proteomics starts as early as the bottom-up method. Recall the protein interference problem, when peptide sequences exist from multiple proteins in the proteome. This causes unambiguous origin determination, right? So if we have a sample with a bunch of proteins and we cut up the proteins using trypsin, like the bottom-up method says, and we might end up with peptide sequences that are the same from different proteins within the sample, so we can't really determine the true origin of these peptides. So the solution to that is computational methods. The first method is parsimony, or what we call Occam's razor. What you're doing is you're creating a list with a minimum number of proteins that accounts for all detected peptides. So you have all your peptides that you detected that you want to analyze, and you're going to go through all the proteins present in the sample, and maybe even then some, and say, which proteins can account for everything here? And then you're going to make that list with the minimum number of proteins. So you don't want to go too over. You're just going to get just the right amount of proteins that account for it. The second solution is the protein profit or algorithm. So this uses a probabilistic PSM score to calculate the probability that the protein is present in the sample. Essentially, that just basically means that it looks for the possibility that this protein can be the origin of that peptide that we see. And the bottom note here says that it adjusts for the number of sibling proteins and same sequences from different precursor charges. Essentially, that just means it adjusts for how many peptides that can be found within the same protein. That's what that basically means. So recall, parsimony is just creating how many proteins can be made of these detected peptides, versus protein profit algorithm, where it's essentially who can donate these proteins or who can donate these peptides and any related peptides to it. Important to note that all solutions, so either parsimony and protein profit algorithm, they are accepted within the proteomic community. There is no consensus on which works best. However, journals, any publishings require the results to be reported based on the parsimonious protein groups. So, academic journals, scientific journals will only accept proteomic studies if the results are presented with the parsimony solution. And the last two challenges, one of them is specific to forensics, but one of them is the incorrect protein spectral matches. So if we're doing our database search and we have incorrect like protein spectra that are just way off, we must remove them, right? And the solution to this, simple enough, it's the threshold. You're adding the threshold, say this much, let's say 70 out of 80% similarity, or 70 out of 100% similarity is good enough to be considered a true positive or a positive. It's stuff like that. So you're applying the threshold to just filter out everything that is truly incorrect. And finally, specific to forensic analysis, there are issues with proteomics as sometimes the sample origin is unknown, which then leads to an unknown contaminant, an unknown standard operating procedure, and an unknown database for the protein identification. However, the unknown contaminant and unknown standard operating procedure, yes, those are challenges, but the unknown database can be overcome. Recall the de novo identification for untargeted data analysis. De novo identification does not use a database. So there have been studies, and these studies have proven that despite having no database, analysts were able to ID the taxonomy of the origin of this, of the species of origin with de novo identification. Finally, we have the case study. So the link is at the very bottom of the slides underneath the title. To give some context, the deceased, Aleka Gonzalez, was found dead in North Vancouver, British Columbia. She was last seen with her babysitter, Henry Thomas, before her death. The initial investigation into her death showed that she had bruises on her body, which led investigators to believe that blunt force trauma was the cause of death. However, bruises were consistent with a bite from a venomous animal, which then prompted a proteomic investigation started by Dr. Leonard J. Foster at UBC. Important to note as evidence that Henry Thomas had snakes as pets. So, while reading the case study, I'm not going to go through it as a whole, but it's just a little side activity if you would like to read it and just try to apply everything that from these past two lectures into the case study to see what you can point out and stuff that you can look at and just connect ideas essentially. Here's some questions to probably prompt that connection forming thing. The first question is, what approach / method did they use? They do specifically state it within the paper. However, you might be able to deduce what type of method or approach they used just by reading it. How many samples were analyzed and how were they prepared? So our sample preparation, they did do things that we did not cover, that I did not cover within the lecture series, but just important to note, how were they prepared? Why did they prepare it? Stuff like that. What instrumentation and specific conditions were used? There's supposed to be a question mark after that. Just ignore that for a minute. So essentially, how did they analyze their data, or how did they analyze their sample? How do they analyze the resulting data? So, which method out of the 5 that I showed you was employed for the analysis? And finally, what interferences or challenges did they have to consider while doing the study? So those are just some questions: the who, what, when, where, why, how on proteomics. Of like, while you're reading, you should consider, these questions so that you can apply the content from these lectures into reading it. And I'll just give you like a little bit of a spoiling ending, so you can mostly focus on the method section, which is where most of the data or most of the information and the answers to the questions really are. Just for some context for the conclusion, 111 peptides from snake venom detected. Only one matched with phospholipase A2, and others were housekeeping proteins. Important to note that there are three main protein families in snake venom. You have phospholipases, serine proteases, and metalloproteases. And no metalloprotease or serine proteases were detected from their analysis, and this was hypothesized to be because they have a short half-life. So metalloproteases and serine proteases have a short half-life, and that's why they were not able to detect the any of the peptides. The peptide found in snake hemoglobin was also found in the victim's blood and in diamondback venom. And finally, ELISA testing was employed for snake venom, which attributed a member of the Crotalus genus as a species of origin. The Dr. Foster and his team, they did, in retrospect, do a retrospective and said the species of origin may have been determined unambiguously had multi-reaction monitoring been employed. Regardless of whether or not the species of origin was determined, Henry Thomas, the babysitter, was sentenced to 18 months in jail. That is all I could find on the case, this one case report. And this concludes the lecture series on forensic proteomics. Once again, it has been Sean Calingayan, and it has been a fun time. See you in the next one, hopefully.